We have established a new protocol for the direct reprogramming of human urine-derived cells into myotubes. This in vitro modeling enables us to evaluate exon skipping in the myotubes derived from patients with Duchenne muscular dystrophy.
We discovered that 3-deoxynitrogen A hydrochloride (DZNF), a histone methyltransferase inhibitor, could significantly promote direct reprogramming of UDCs into myotube in a short amount of time. This new autologous UDC-based disease modeling could lead to the application of precision medicine for various muscular diseases.
To begin, centrifuge the entire urine sample at 400 times G for 10 minutes at room temperature. Next, aspirate the supernatant, leaving 1 milliliter in the tube. Resuspend the pellets individually in the remaining 1 milliliter of urine. Collect those in a single 50 milliliter tube and add 10 milliliters of washing buffer to the tube. Centrifuge the samples at 200 times G for 10 minutes at room temperature. Aspirate the supernatant, leaving 0.2 milliliters in the tube. Resuspend the cell pellets in 4.5 milliliters of primary medium. Seed the cells in 3 wells of a gelatin-coated 6-well plate, 1.5 milliliters of cell suspension per well. Culture at 37 degrees Celsius and 5% carbon dioxide. For 3 days, add 1.5 milliliters of the primary medium each day. On day 4, replace the medium with 1.5 milliliters of growth medium